Ladies and gentlemen, I would like to introduce here well-known American researcher, Professor Richard from NASA. Professor Hoover works in astrophysics and astrobiology. He investigates icy satellites, organic planets, meteorites, other bodies. He is also of more than 400 scientific articles, monographs, books, editions. He is also of 15 inventions in optics and photonics. Professor Hoover investigates problem of microfossil meteorite substance. He was awarded with a gold medal of International Society of the Optics and Photonics. Professor Hoover, please.
Thank you very much, Professor. I am most honored to be with you here today in Tbilisi. This is my first visit to Georgia, and I have found your city to be extremely beautiful, and this has been a wonderful visit. I'm talking today about the study of microfossils in meteorites. I have started the work on the study of microfossils in meteorites almost a quarter of a century ago. Astrobiology is a very broad and multidisciplinary program which is devoted to the study of life in the universe. Astrobiologists are involved in all manner of things, from the study of planets to the study of microorganisms and meteorites and terrestrial rocks and a wide variety of other fields. The fundamental question of astrobiology is life restricted to the planet Earth? The current paradigm, of course, in biology has been that all life on Earth originated by abiotic processes on the primordial Earth, basically by the process of lightning bolts and volcanic eruptions causing the formation of amino acids and other organics that rained out in primitive oceans and seas, and then after a long period of chemical evolution resulted in the formation of the first protocells that then evolved into all the life forms that we know on Earth. However, recent discoveries of microfossils and biomolecules in meteorites and comets and of heat-producing elements in deep crustal rocks indicate that life is widely distributed throughout the cosmos. One of the fundamental problems with the origin of life on Earth, protocells have never been detected in any of the ancient rock record study. The other problem is that the idea that these complex organics were first formed and then developed into simple cells is that simple cells just do not exist. We have here an image of the Dna Gyrase enzyme, and you see all the coils and folds that go into the Dna Gyrase enzyme of E. coli. Here is a bacterial motor, a transmission electron microscope image of a bacterial flagellar motor, and you have the rotors and stators. This is like something that could have been invented by someone like Nikola Tesla. However, it was actually invented by bacteria some 4 billion years ago on the planet Earth. The human genome contains only 3.2 billion base pairs, whereas this strange little amoeba, which is known as Polychaos dubium, contains 670 billion base pairs. And the fossil record of Earth indicates that life appeared somewhere between 3.8 and 4.2 billion years ago. That is about the same time that liquid water oceans began to form on the planet. Sharoff did a study of the timeline of the origin of life by linear regression of genetic complexity, looking here at mammals and fish and worms coming back to the first eukaryotes and prokaryotes. It looks like that in this linear regression indicates that the origin of life on Earth, which would have been here 4.5 billion years ago when the Earth formed, that life had to have been developing for a very long period of time before that, and that means, of course, if that is correct, that life had to have originated somewhere other than the planet Earth. All life forms on Earth require the coexistence of three things. You have to have water, you have to have an energy source, and you have to have a small group, 20 of the 118 chemical elements. These are called the bioelements. The second most abundant molecule in the universe, and by far the most abundant, in which you have two or more different elements, is the water molecule. It comprises some 65% of the mass of every living cell. It has a polar bent geometry with this hydrogen-oxygen-hydrogen bond angle at 104.5 degrees. That is the reason that water has such unique properties. It expands on freezing, it has a high surface tension and boiling point. Well, because of this very, very important characteristic of water, when it freezes, the crystallization process results in the frozen water having a lower density than the liquid water. That is wonderful, because if that were not the case, none of us would be alive. The reason that that is because when the water freezes, the ice floats to the surface, not just in your martini glass, but on all the seas and oceans and lakes and rivers of the planet Earth. That means very definitively that underneath this polar ice sheet, there is liquid water. And the interesting thing is that water freezes at plus 4 degrees Celsius at the seawater, plus 3.9 for fresh water, and minus 0.8 to plus 2 for seawater. Actually, there's a region called the Antarctic bottom water, and that has a temperature in that regime, and that is found throughout all of the polar regions of the planet Earth. The fascinating thing about that is that means that all water oceans everywhere in the universe would be alike. This is the the comet 9P Temple 1. Comet 9P Temple 1 was studied by NASA. This is an image of 9P Temple 1 when it was out beyond the orbit of Mars. And you see that because of sunlight striking the dark region where the sun is shining, it gets quite hot. This is showing the upper temperature of 330 degrees Celsius. And this whole region back here is shown in 280 to 290. Well, 273 Celsius is the magic number. That is the temperature at which, 273 Kelvin is the magic number. That is the temperature at which water freezes into ice. 330 Kelvin is indeed quite warm. And most of this region of the comet is at 0 Celsius, which is the temperature in which the ice underneath the rocky organic crust would have melted. This data here shows evidence from the mission of water ice absorption bands indicating that there is indeed water ice associated with this comet surface. Here are jets being produced by 67P Trichome Gyrase Enzyme, which was a magnificent comet that has been studied extensively very recently. And interestingly, as the jets come up, they carry with them dust and particulates as well as water that freezes. And there is a video of snow on the surface of 67P Trichome Gyrase Enzyme. In 2018, Professor Shimonia and Dale Cruikshank of NASA Ames published a very, very interesting and very important paper in which they were able to match the spectral lines. This is Comet de Vico, and they got very, very nice matches between the spectral lines that had been unidentified in the comet with enzymes like luciferase, with chlorophyll A and chlorophyll, with the green fluorescent protein. That is a profoundly exciting result. In fact, that's the reason that I'm here in Tbilisi today. I was unaware of this paper until just about a month ago, and after I saw it, I was very, very much struck by it and called Professor Shimonia and realized that I needed to come and have a chance to talk with him about possibly maybe even undergoing future studies with regard to this important work. The fascinating thing is this is the enzyme Luciferase, and luciferase is not like a simple organic like ethyl alcohol or methane or formamide. Luciferase is a very, very complex biomolecule that is formed by the determination of the genes that tell precisely where every one of 20 amino acids has to be placed, and a luciferase molecule contains 526 amino acids, each one at a precise location within the enzyme. The fascinating point of all this is the number of atoms in the universe is calculated to be approximately 10 to the 80th. If you took a soup of amino acids and said, let us at random at each point pick one of the 20 amino acids, in order to make the luciferase molecule, you would get it right in about 1 out of every 20 to the 526th power times. That is vastly higher than the total number of atoms in the universe. Many years ago, academician Eric Galimov was studying meteorites, and he was actually studying the biological fractionation, and in his book he showed where in the Murchison meteorite the biological fractionation of glycine, glutamic acid, alanine, and valine was very, very similar in their relationship to Euglena glacialis and some other microorganisms like chlorella. But the fascinating thing is this is the carbon isotopic fractionation, and in living things you see the del 13C goes from about -4 to around -24, but the Murchison meteorite has extremely heavy carbon, so you see here the fractionation of those amino acids goes in the same way, but along a very, very different scale from +20 to about +50. When I saw that in academician Galimov's book, I was shocked and told him that it looked to me like the best geochemical evidence for the existence of extraterrestrial life. He smiled and said, yes, I think I can see how you might interpret it that way. Of course, he had simply said that it was an analogous relation, but I think he really realized it was indeed indicative of biology. The energy can come from a variety of sources, and life is characterized by metabolism, DNA and RNA-driven ordering, chemical complexity, growth, reaction to stimuli, reproduction, and motility. One of the fascinating things is that living organisms such as bacteria and diatoms and algae, when they swim through water, because of the size of these organisms, the physics of this locomotion is governed by the Reynolds number. And when you look at the Reynolds number for organisms the size of bacteria swimming in water, it would be the equivalent of a human being trying to swim in an amorphous substance like glass. So an enormous amount of energy is expended in bacteria as they swim through water, and that means that observing the swimming motions of organisms on Mars or on comets would be one mechanism in which you could determine between whether something you were looking at was a living organism or was particles that were being drifting or are being moved by simple Brownian motion. The living organisms require 20 bioelements. The major ones are CHONPS. Actually, about 98% of all life is carbon, hydrogen, oxygen, and nitrogen. And fascinatingly, the interstellar component of cometary dust is made of cone particles, carbon, hydrogen, oxygen, and nitrogen particles. This is another evidence indicating biology in comets. One of the fascinating things of life is all living things are use homocural biomolecules. The amino acids and sugars are made in which the chemical composition is identical, but the molecules themselves exhibit chirality. That is, you have some that are like left-handed gloves and some that are like right-handed gloves. The left-handed biomolecules rotate the plane of polarization of visible light in the levorotary direction or counterclockwise direction. The right-handed ones in the dextrorotary direction or the clockwise direction. If you make these kind of molecules by chemical processes, you always get racemic mixtures, which means the same number of the D molecule as the L molecule. So in racemic mixtures, D over L equal 1. This shows an L-alanine and a D-alanine, and even though they are chemically the same compound, they have this very different structural configuration. The Orgay meteorite contains only 8 of the 20 protein amino acids, some of which have a significant excess of the L enantiomer. All life on Earth, all proteins, all enzymes are made strictly of the L amino acids, and all of the sugars in living things are made strictly of the D sugars. We know from the Watson-Crick canonical base pairing that in DNA, guanine pairs with cytosine and adenine pairs with thymine. In RNA, the guanine pairs with cytosine and the adenine pairs with uracil. So the fascinating point is that that means that in every living organism, you have exactly the same amount of guanine as you have cytosine. Well, one of the fascinating things is that in the carbonaceous meteorites, thymine and cytosine have been found to be absent until just recently, when guanine and when cytosine and thymine were discovered in the Murchison meteorite, but they're 2 to 3 orders of magnitude lower in content than their relative paired nucleobase. In 2008, Martins et al. Proved that the nucleobases in the Murchison meteorite were extraterrestrial by measuring the del 13C of uracil and finding it to be plus 44, which clearly is proof this could not be a terrestrial biological contaminant. The Murchison meteorite fell on May the 14th, 1864 at 8 o'clock, and when it came in, it was a gigantic fireball that caused all kinds of consternation. The people thought that there was an earthquake. Some thought the world was coming to an end. In 1962, George Claus and Bartholomew Nagy detected microfossils in the Orgay meteorite, but they were criticized so heavily, they said, well, we don't know exactly what these are, so they started calling them organized elements. In 1963, however, the Hungarian botanist Palik reported the discovery of these kinds of filaments in Orgay, and she called them blue-green algae. We now know that blue-green algae are not algae at all. They're cyanobacteria. They're bacteria rather than algae. But the fascinating thing is the cyanobacteria can be quite large compared to other bacteria, because you have many bacteria, many of the bacterial cells that inhabit sheaths, and these chains of cells are called trichomes, and sometimes they form specialized cells. Like at the top of this image, she showed this little structure here. This is an image I took with the scanning electron microscope, and there is the same kind of structure on this tapered filament, and that structure is called a calyptra. Inside the cells, inside the filament, this particular cyanobacteria reproduces by what is called trichome fission. The chain of cells breaks and small chains come out into the water, and here you have a small chain, a small trichome. This is called a hormigonium, and you can see here 1, 2, and 3 cells on the inside. Now the important point is here we're seeing Microcolia stonoplasty, we're seeing evidence of formidium and evidence of nostoc, which is a community that lives together in polar regions, and they're in the Murchison CM2 carbonaceous meteorite. We have good evidence from the studies that were done after the report of the trajectory, as people reported that they saw the bolide that made the Orgay meteorites at this point in the sky when they were at that location, and at this point in the sky when they were at that location. And by using that, scientists and astronomers were able to determine a trajectory of the meteorite fireball, and then they were able to locate and collect samples of the meteorite from all along the path. That led to the conclusion that Orgay is either from a Halley type or a Jupiter family comet, and we now know that liquid water existed on the parent body of that meteorite as recently as 10 million years ago. In 1997, after David McKay made his announcement of possible microfossils in the Orgay meteorite, I started doing a study because I thought everybody knew there were fossils in meteorites. I didn't realize that this was at all a controversial topic, and I didn't realize that NASA would get all excited about it, because I had known about it for years, but since I had followed the work in the early 1960s. Well, I thought, wait a minute, why is everybody upset? Why do they really exist? We had at NASA Marshall Space Flight Center an environmental scanning electron microscope. Here you see here, and this is academician Alexei Rozanov when he was at that time director of the Paleontological Institute of the Russian Academy of Sciences in Moscow. He was then a young boy, and he was just a corresponding member of the Academy at that time. Subsequently, for his work on microfossils and meteorites in 2008, Alexei Rozanov was elevated to a full member of the Russian Academy of Sciences. The picture here is some of his images that he did in a paper in 1997. This is images of a paper that I did in 1997. We independently published the discovery of microfossils in the Murchison CM2 carbonaceous meteorite, and then we worked collaboratively and studied fossils in ancient rocks. These are in the Cambrian deposits of Hubsagul, Mongolia. So we see here this chain of cells, and here very similar is a formidium here, and you see an apical calyptra here. This one is broken there, but embedded in the rock, you see these calyptra that are sticking up and are just at the surface of the rock. Here is a chain of an emergent trichome from a Nostocacean cyanobacteria, and here is where the filament is broken, but you can see it's hollow. And over here, you can see cross-wall constrictions where it's flattened, and that makes it possible to determine the exact size and shape of the cells, and we can identify this particular species of nostoc. When we put the electron beam here and measure the elemental composition, in this hormigonium, we can actually detect nitrogen, but it's only 0.2%. Almost always in these fossils, we are never able to detect nitrogen. But here the carbon is 41% and the oxygen is approximately 16%. The meteorites we have now found contain not only cyanobacteria, but a wide variety of other forms, including, most excitingly, diatoms. A lot of people like to criticize and say, can you really tell that those are really biological things? Well, if you don't understand and you don't know cyanobacteria, you might not recognize them, and you might suspect that they maybe could be made by abiological processing. When you look at the incredible magnificence of the structure of diatoms, no one, even a non-biologist, is ever going to look at those and say, I don't think those things were made by biology. I think they were some kind of chemical fluke. Well, diatoms have long been considered to be phototrophic algae that inhabit, and they do inhabit, the deep oceans. They inhabit deep layers of the oceans as well as the upper layers of the ocean. They also are the most abundant life forms in Antarctica. And when it goes dark in Antarctica, it gets really dark for about 9 months. And in the Antarctic oceans, in the Antarctic ices, the diatoms can live and can go about their life by producing enzymes that melt the ices, so they're actually living in water. And they can also eat organic chemicals, and one of the amazing things is they can live on alpha-aminoisobutyric acid, which is known and an abundant amino acid in meteorites, but it is not known on Earth except when it has been delivered by meteorites. Bacteria, this is an image from Lake Untersee taken by Dale Anderson when he dived under 10 meters of ice and 30 meters of water. And you see here these stromatolites on the floor of Lake Untersee are deep red in color. Just 4 years ago, Fuente Sanchez et al. Published a paper which was also very, very important about the existence of viable cyanobacteria in the deep continental subsurface. They found cyanobacteria deep in the rocks under Rio Tinto, and the amazing thing is those cyanobacteria were not carrying out photosynthesis. They were instead doing hydrogen metabolism. They were eating hydrogen that was being produced by radioactivity in the deep crustal rocks. In 1986, I had the great opportunity to work with the brilliant genius Sir Fred Hoyle. Fred Hoyle, as you may know, was the one who in 1946 described how hot stars provide the mechanism whereby nuclear fusion within stars can give rise to the different chemical elements. This process, known as nucleosynthesis, was pioneered by Fred Hoyle, and in that work he showed the mechanism whereby carbon could become in a resonant state and become stable. That came to be known as the Hoyle state. It occurs every 1 out of every 2,500 reactions in which 3 alpha particles combine. It has been called the most outrageous prediction in the history of science because within the last decade it has been possible to prove not only theoretically using supercomputers but also using synchrotrons that this Hoyle state does indeed exist, and by the emission of a 653 MeV gamma ray, just as Fred Hoyle predicted in 1946. We started working on the problem that he and his student, Professor Wickramasinghe, had, and that was in explaining the interstellar characteristics of interstellar dust. The interesting thing is that the diatom builds a shell, which is a hydrous amorphous polymer of silica that is made in this way. You have a cellulose template and a protein template, and that causes the form. These are the amino acids dictated by the DNA that tell the diatom exactly to where to put each molecule of silica, and the silica is actually SiO2 times NH2O. So it is a hydrous biopolymer. The problem is if you look at sand grains or other crystalline rocks, inorganic rocks, the infrared spectra shows sharp peaks at 8.7 and 12.7 microns. Here is the spectrum of the Trapezium Nebula, and you see it matches very nicely to the spectrum of diatoms within the infrared between 8 and 32 microns. Here is the infrared source at the galactic center, GCIRS-7, and all of these nice little bumps and wiggles are matched by the astronomical data and the data that we measured at the Naval Research Laboratory when Sir Fred Hoyle came and visited me at NASA. He gave a wonderful lecture there, and then we went on to the Naval Research Laboratory in Washington to obtain this data. One of the interesting points is that a lot of people think that, okay, if these structures are in the meteorites and they're really biological, they have to be modern contaminants. Well, if the things we're finding in the meteorites are modern biology that crawled into the meteorite after it landed, it means nothing of significance whatsoever. If they were in the meteorite when it came into the atmosphere, it is proof of the existence of extraterrestrial life, which is indeed vastly more significant than bacteria crawling into a rock after it landed. Well, I spent many years trying to figure out how could one absolutely show conclusively that the structures found in the meteorites were indeed fossils rather than modern material that had crawled into the rock after it landed. And in fact, when we examined these stones, we found that the fossils had a spectra in which you almost never detected nitrogen. And here is a woolly mammoth guard hair. I collected woolly mammoths when I was working in the far northeast of Siberia in the Kolyma lowlands. I have in my freezer at home heart, lung, tissue, and milk of baby mammoth Liuba. And here we see where the electron beam is. Is it possible to lower the lights? Maybe it will be easier to see. Well, perhaps not. Here is where this beam struck the hair, and you can see here a very beautiful nitrogen peak and carbon and oxygen. Carbon is much higher than you would expect it to be, but that is because this hair is 32,000 years old and it started to go into a form of humic acids and then ultimately to diagenetic materials like coal. This is not a hair of a modern woolly mammoth. And here we see the spectra, the data on orgate filaments, and here is a contaminant on a piece of Murchison that I had, and these are living cyanobacteria. These are from pre-dynastic mummies from Egypt and mammoth hair and tissue, and these are from Cambrian trilobites and Precambrian cyanobacteria. This shows a filament that has been converted entirely into magnesium sulfate. It has fossilized as magnesium sulfate on the interior of the Orgay meteorite. The spectrum taken here shows no carbon at all, very high magnesium and sulfur and oxygen. Magnesium sulfate is water soluble. It's also known as epsomite. If this had been subjected to water after the meteorite fell, those forms within the meteorite would be totally dissolved, and therefore we would never be able to see them. In the meteorite, we also find these beautiful filaments like this with a terminal hair. This is a very large cyanobacteria. That's a 5-micron bar. And at this spot here, the carbon had almost 82% and the oxygen 9%. That is a chemical composition totally different from biology, from living biology. It is a chemical composition such as you would expect to find in anthracite coal. In 2017, at the Joint Institute of Nuclear Research in Dubna, we made the first discovery of diatoms in the Orgay meteorite. These diatoms died shortly after they reproduced by mitosis, and one can see here that this one is very, very slightly smaller than that because of the way diatoms replicate. And in fact, it was possible to identify this diatom as being very closely allied to the diatom Pinularia segeriana that was discovered in 1955 by Neil Foged, and he found that in New Zealand. We published the discovery paper in 2018 in the Paleontological Journal. There are several other diatoms that we've found now in the Orgay meteorite. Here are two others that are seen nestled in the meteorite. This is possibly an Ecnanthes, another diatom, and fragments in the meteorite. In 2012, a huge fireball was observed over north-central Sri Lanka, the Polonnaruwa district, and several of these stones fell in near the vicinity of the village Aralaganwilla. This is one that I collected while I was there exactly 1 month after the fall. We have the oxygen isotopes that show that these stones are far away from what's called the terrestrial fractionation line. All rocks on the Earth and on the Moon fall on that terrestrial fractionation line, proving conclusively that the stones are not terrestrial rocks. Furthermore, they contain fractured grains of ilmenite and olivines and zircons, and this strange mineral called mascalinite. Mascalinite is a pleomorph of plagioclase that is found about 20 kilometers down, 20 to 40 kilometers in the Earth's crust, and it forms only at shock pressures of the order of 20 gigapascals. These are beautiful diatoms from within the Polonnaruwa meteorite. Here you have a beautiful long chain of Alococera ambigua. This is an enlargement, and here is the same diatom from a terrestrial sample. In the meteorite, it is completely covered with the gelatinous sheath, which is almost never seen in terrestrial fossil material. And here's another diatom from Polonnaruwa. This one is found in the Aralaganwilla Polonnaruwa suite, and this was found by Chudayev and described from the Voronezh region of Russia in 2020. So here we have exactly the same type of diatom, probably same genus, maybe even same species, seen in the Polonnaruwa material. These are two others, or three others actually, one with a nice square set of pores. This is a very strange object that I believe to be a hystricosphere with long spines in the Polonnaruwa meteorite, and acrotarchs in Polonnaruwa. In 2016, Dr. Marina Frontisyeva and I started doing neutron activation analysis of the meteorites, and these are standard carbonaceous chondrites going from here down to here, and then the Polonnaruwa, Aralaganwilla meteorites are seen here. And one of the things that is most interesting is if you look at the potassium content, 130 to 140,000 part per million, as opposed to 600 part per million for the other carbonaceous meteorites. The uranium and thorium are also extremely high. This completely separates these Polonnaruwa Aralaganwilla stones from all of the other meteorites. Another thing that separates it is they have a density of 0.6 to 0.8 and 60 to 80% porosity. This kind of porosity is only found in pumice and in diatomite, and it has also been recently discovered in dart boulders from asteroid Bennu and Ryugu. Deep in the crust of the Earth, there is a biosphere which is fed by the radioactive decay of potassium, uranium, and thorium. It turns out that when they decay, they give off gamma rays and antineutrinos, and we now know from the Ice Cube Antineutrino Telescope in Antarctica and some of the other antineutrino telescopes that the Earth is producing far more antineutrinos than it is receiving. So in fact, from an astronomical point of view, the planet Earth is an antineutrino star. The other thing that is very, very exciting is that deep in the crust of the Earth, there are no oceans, atmospheres, or sunlight, and yet there is still life. So oceans and atmospheres of sunlight, which many planetary scientists have been searching for planets like Earth because they are looking for planets with oceans. I'm sorry, you do not need oceans for life. You also, when you do have oceans, you can carry out this phenomenon of serpentinization. Tommy Gold predicted the deep hot biosphere many years ago. He was a bit ridiculed for that, but it turns out that despite the ridicule, Professor Gold was correct. There are two processes in the formation of serpentine and magnetite, both of which are found in the carbonaceous meteorites, and both of which are exothermic processes. So that is another mechanism where you could provide heating for liquid water oceans underneath ices. I've spent many decades going around the world to Antarctica, to Siberia, and so forth, studying life in ice. This is the glacier, the Matanuska Glacier of Alaska, the Shermaka Oasis ice cube, and here is a glacier in Iceland and in the high Alps of Austria. And this is me with the astronaut Jim Lovell and Owen Garriott during our astrobiology expedition 2000, and that picture was taken at the geographic south pole of the planet Earth. In Lake Untersee, we discovered an entirely new family, new genus, and new species of bacteria, which has these strange structures that are new organelles that we called antii. Here are bacteria that were brought into water from the tissues of Liuba. This mammoth died and was drowned some 32,000 years ago, and yet Dr. Pakuta and I were able to culture these organisms from the tissues of Liuba. With Sabita Buizov, who was the first to predict biology in the deep ice of Antarctica. These are diatoms from the 2,827-meter core of Lake Vostok, Antarctica. Very, very beautifully preserved diatoms. They look in the same kind of vein as the type of preservation that we have found diatoms in the meteorites. This is a living organism, a small protozoan, living and swimming in a piece of ice that I collected in the Whistler Ice Cave of Canada in 2016. And one of the amazing things is in that ice cave, we also found some beautiful and spectacular ice worms. These ice worms can be seen crawling through the ice, and if you look at them under the electron microscope, this beautiful little ice worm, well, maybe he would be beautiful to another ice worm, or she. The interesting thing is the polar caps of Mars contain water ice. It's about 3.6 kilometers thick at the North Pole of Mars, and then it is covered over by a 10-meter thick layer of carbon dioxide in the winter. So this means that in the polar regions you have a vast amount of frozen water. One of the things that we're hoping to do with SpaceX in the future is fly a mission to Mars, in which we are hoping our experiment that we call the Twin Wireless Extraterrestrial Experiment for Life, named TWEAL, is to collect samples of the Martian regolith by sending little arrows out with a penetrator that smashes into the Martian surface and ruptures these ampules and releases nutrient media. In one half you have L-aminos and D sugars. In the other side you have D-aminos and L sugars. And when you get the response curves, if you have two identical response curves, it would be proof it was chemistry. If you have one where the L-aminos and D sugars respond and the others don't, it would be life like Earth life, and the opposite would be biology that is entirely different than Earth life. This is what we hope will be our vehicle to take us to Mars. This is the magnificent Starship 2 that Elon Musk has built. This is not a drawing, not a painting. This is a real photograph of Starship 2 sitting on the launch pad, ready to be launched. And one of the interesting things is that underneath the ice crusts of many moons and planets of our solar system, there are ocean worlds. We have ocean worlds that are now known to exist underneath the icy moons of Jupiter, like Europa and Ganymede and Callisto. And here you have a rocky, a molten core, then a rocky area, then a liquid water ocean, and then the exterior icy crust. Enceladus also is showing this sort of thing, as well as the tiny planet Pluto. One of the intriguing things is that on Europa, you see these deep red lines. This is cracks in the surface of Europa. Here is the Connemara Chaos region, showing the golden browns, the white snow, the blue ice, and these golden brown regions. This is snow algae that I collected in Siberia, and here again are the cyanobacterial stromatolites from Lake Huntersay. This is when the Cassini mission went to past the planets. As it passed by Enceladus, it saw this beautiful ejection of water coming out of the tiger stripes of Saturn's moon Enceladus, proving that there is indeed a liquid water ocean underneath the icy crust of Enceladus. The OSIRIS-REx has been to asteroid Bennu and did sampling of Bennu, and there on the surface of Bennu it discovered this large boulder called Gargoyle Saxum. It has a porosity of 70 to 80% and density of about 0.6 to 0.8. It is almost identical in physical characteristics to this piece of the Arlaganwilla meteorite, which has a density of 0.6 grams per cubic centimeter and a porosity of 80%. The outer surface shows the fusion crust and inner region that I have broken. We did calculations, even though Rositas in his paper said these weaker boulders wouldn't survive atmospheric entry. We did calculations and published a paper in 2013 showing that indeed such low density bodies could enter the Earth's atmosphere successfully. Transplanetary contamination is one of the possibilities of a way in which biospheres could have arrived on Earth. Captain Star is not extremely far away, and in fact, this object has a planet that is 11 billion years old, and it was some 7 light years away from Earth 8,500 years ago. The transit time is only like 85,000 years. Comet 2I Borisov came in a hyperbolic trajectory like this through our solar system. It had a perihelion of 2 AU on the 8th of December 2019. This very exciting interstellar comet contains water and carbon, and it was at Kruger 60 about 1 million years ago. There are possibilities of rogue planets and exomoons that could be dislodged from their solar systems and travel throughout deep space, carrying with them a cargo of biology. The fact that Pluto has a liquid water ocean, even though it does not have a nearby giant star, indicates that oceans could remain liquid for periods of billions of years, and within those oceans there could be entire biospheres, such as the Hadal biosphere of the planet Earth. So this is a mechanism whereby there could not be just interstellar transfer of biology, but perhaps even intergalactic transfer of biology. Rogue planets and exomoons provide a new kind of panspermia hypothesis, and they could explain the rapid appearance of complex life on Earth. The one point that this does, it doesn't explain where, when, or how life originated, but it dramatically alters the time period available for prebiotic chemistry and molecular or cellular evolution. And indeed, biology living within deep crustal rocks of extraterrestrial planets and moons could also survive the transit from one distant place to another. In conclusion, the conditions for life, water, and energy and biogenetic elements coexist on asteroids, comets, planets, and moons of our solar system. There is present indigenous water, microfossils, and ancient biomolecules, including pristane, phytane, nucleobases, and L-amino acids in the Ci1 carbonaceous meteorites. The absence of DNA and RNA, the absence of many of the protein amino acids, the absence of thymine and cytosine, and unusual carbon-nitrogen and carbon-oxygen ratios absolutely prove that these meteorites are not contaminated by modern biology. The infrared and UV spectra of trapezium and the GCIRS center source indicate silica biopolymers, and the comet visible light data suggest even proteins, luciferase, and chlorophylls, which are very, very complex biomolecules. Therefore, in conclusion, I say to you, extraterrestrial life exists. Biospheres in ice-covered oceans or hot subcrustal rocks and comets or rogue planets may be distributed throughout the entire universe. And the fundamental paradigm that all life on Earth originated on Earth may simply not be valid. And with that, I thank you very much for your attention, and I'm now open for any questions you might have.
Is supported in biology here. Why were we unable to find the protocell? The reason is the protocell didn't contain any solid structures, so it has simply has been decomposed, and we cannot find any evidence of it. However, in the modern cell, we do have two structures which have independent genetic material, which have their own DNA, and one is mitochondria, and the other one is chlorophyll, chloroplast, which might have originated earlier, before the protocell, and then have been incorporated into a more complex cell. So this is the biological proof of one of the aspects of the origin of life on plants. However, of course, the biology doesn't deny that the organic molecules which have originated in space indeed have contributed to the life on the Earth in terms of biodiversity or bringing more genetic material to the planet. And what do you think about that? Could this be a process which involved both events, which is abiogenesis in the oceans and by the Müller-Urey experiment, has been proved by Müller-Urey and kind of experiments, and also the organic material brought from the space by meteorites, or rather, separately, one or another?
One of the fundamental problems with the origin of Earth life on Earth, and that fundamental problem is that homochirality is a signature of life. There have been many, many scientists for the last many decades that have been trying all manner of mechanisms in which to show how you could get a homochiral suite of amino acids by abiotic processes using amplification techniques, for example, with crystallization, using techniques of circular polarization of light. Of course, the problem with circular polarization of light happens around neutron stars, which are not close to the planet Earth. So the problem of biological homochirality is indeed a profound problem for the origin of life by chemical processes within the Earth itself. So the other, I recognize that biologists advance the argument that, well, protocells wouldn't be fossilized because they don't have the same kind of structures. But in fact, no biologist has ever been able to make a protocell to show you what it really does look like. So, you know, there have been studies like Dave Deemer with coacervates and so forth, and there have been a wide variety of others. But the fundamental problem is it clearly looks like to have life, you have absolutely got to have this ordering process associated with DNA and our RNA. So now you're back to the formation of incredibly complex molecules. There is no simple DNA. There is no simple RNA. And in order for that to have occurred on the planet Earth, we now know that it would have to have taken place within an onion skin layer of 100 million years or less. And that was the idea that the origin of life was a very quick, simple, easy process. But during the last half a century, the work has been done on trying to explain this quick, simple, easy process. It's gone the other direction. We now know that every living organism is incredibly complex. And the difference between a bacteria and a human neuron is vastly different than the difference between a grain of sand and an E. coli bacteria. So my argument is that the fossils indicate that there was biology that was existent other places in the cosmos, and that the amount of time for this chemical evolution and the mechanisms whereby homochirality originated probably were vastly longer than anything that would have been available on Earth. So that's why I know this, what you have cited, is the fundamental paradigm of life that is accepted by biologists all over the world. And that is one of the problems with the discovery of fossils in meteorites, because it challenges not just the very fundamental paradigm of the origin of life on Earth, it challenges all sorts of things, such as, does life exist elsewhere? And in fact, if we take that hypothesis, that life exists on the planet Earth, and we know it exists on Earth and essentially everywhere, from the high stratosphere to the deepest regimes of the Haddle zones of the oceans to deep within the crust of the Earth, there is biology. And we pose that against the other part of the hypothesis that life exists nowhere else in the universe. That would be far more astonishing than the hypothesis that existed for several thousands of years, that the Earth was the center of the universe and all of the planets and stars and other objects of the cosmos revolved around this pale blue dot. So what I'm suggesting is that the panspermia hypothesis, whereby biology was probably brought to the planet Earth, is far superior to the endogenous origin of life on Earth.
Thank you very much. Anyone has any questions? Otherwise, I do have another question.
Yes, please.
The other remark from biologists is that the fact that you find fossils in the meteorites, it is considered the not very reliable source for proving panspermia. The first reason is, in the past, the planetary bodies such as asteroids and comets have been falling on Earth. They could have fractured some kind of rock containing cyanobacteria, or the part of the ocean could be taken to the space because of that hit. And then these rocks went to the orbit, and after a long time, they fell back on the Earth. So they could have carried the bacteria which have originated earlier on the Earth, and then now have been brought back to the surface of the planet. So this is why biologists think that this is not a very reliable source for saying that...
the argument they are making is that the meteorites were ejected from planet Earth, went into space, and then came back and brought terrestrial biology with them.
Yes.
That is an argument that biologists make.
Yes.
The problem is, just like planetary scientists making biological arguments, the problem with biological scientists making astronomical and astrophysical arguments is equally the same. They're out of their own realm. Why do I know for a fact that the Orgay carbonaceous meteorite did not come from Earth, and it is very simple. It has been known for a long time that the elemental abundances of the Orgay meteorite, in fact, all of the carbonaceous chondrites, but particularly the CI1s like Orgay and La, the elemental composition of those meteorites, if you look at the content, not considering the volatiles, but the content of all the different elements, they precisely match the photosphere of the sun. They do not match the crust of the planet Earth. And in fact, that proves that they came from the protosolar nebula condensate, which means that the Orgay meteorite, the La meteorite, all of these CI1 carbonaceous chondrites did not come from anything at all related to the planet Earth. In fact, all of the rocks on the planet Earth and all of the rocks on the moon have oxygen isotopes that lie on what is called the terrestrial fractionation line, and all of the meteorites, well, some are fairly close to the Earth's terrestrial fractionation line, and of course we do have lunar meteorites that lie on the terrestrial fractionation line, and tektites, which were things that were blasted off the Earth, went into space, and came back. But those are Earth rocks, and we can recognize those Earth rocks by their oxygen isotopes. But these meteorites have dramatically different carbon isotopes, dramatically different oxygen isotopes. It's possible to measure by cosmic ray exposure ages how long they stayed in space. So it is clearly, definitively provable that the hypothesis that Earth life was blasted into space and then came back on a terrestrial rock is demonstrably false. However, it is possible that Earth life could have been blasted into space, and while orbiting or being in fragments in distant regions, as comets came through and swept up terrestrial ices that maybe were blasted off the polar regions, or when we had a snowball Earth, they could have conceivably accreted terrestrial biology, and when they go close to the sun and produce aqueous environments, then that biology could have conceivably started to grow. But that would be a case of Earth delivering biology that would then live on a parent body and then come back to Earth. Well, the problem, of course, with Orgay and with Polonarua and some of these others, we find some things that are very readily recognizable as terrestrial life forms, but we also find things that no one has been able to identify, bizarre structures that don't look like any kind of organism that I've ever seen. And I had an opportunity at the Komarov Botanical Institute last fall to be sitting around a table with a number of very distinguished scientists whose specialty extended far beyond my specialties as far as dinoflagellates and other kind of microplankton are concerned. And when the director of the institute looked at one of these strange things and I asked him what it was, he said, something biological. And I said, but is it eukaryote or prokaryote? Something biological. The problem was he had no idea whether this was a eukaryotic life form, a prokaryotic life form, what genus, what species, nothing. And I've experienced the same thing. Many of these things simply are not identifiable. So again, I think the problem is very simple. The panspermia hypothesis was discarded early on, and it was discarded before many things were known that are now known. It was thought that they were talking about seeds from space, individual bacteria traveling through space, and then the argument was, well, the radiation is going to wipe them out. Well, they couldn't possibly remain alive for millions to hundreds of millions of years. But if you have a biosphere in which there is biology contained within Pluto, for example, or Earth, if a star came by our own solar system and by some chance came so close that the Earth was ripped out of its orbit, came closer to the sun, perhaps a little bit farther than the distance of Mercury, went swooping around the sun, and then got gravity assist like Pioneer did, from the gas giants Jupiter and Saturn, and it achieved a velocity of 38 kilometers per second. It would then have escape velocity, and planet Earth, as it went out to the far distant regions of our solar system, all the gases of the atmosphere would solidify and condense on the surface of the planet, the water first, then methane and nitrogen and oxygen, and all of these gases would form a crust. But deep in the oceans, there would be these long-lived heat-producing elements, thorium-232 with a half-life of 14.2 billion years, uranium-238, 4.5 billion, and potassium-40, 1.2 billion. So the Earth could go merrily along, whales probably dead, humans and dogs and cats dead, but bacteria, deep-sea hydrothermal vent worms, all manner of crinoids and crabs and shrimp, and all of these deep-sea fishes might be perfectly happy and content on a planet Earth as it hurtles toward the Andromeda Galaxy. Okay. So I give you another kind of starship, our own planet.
All right, thank you very much.
Thank you.
So unless anyone has a question.
I would like to know, well, physically, in physics, and subatomically, everything is made of, atoms and those particles which atom contains. And I would like to know if those microorganisms have the capability to evolve and create some kind of like society as humans, well, time to time.
I find little blue-green algae, I don't really know anything about little green men, but several years ago we had a symposium that I organized in San Diego, and we had a live video feed with Sir Arthur C. Clarke, very famous scientist, and a young boy about 8 years of age came to the microphone and said, "Professor Clarke, what do you think are the chances of finding intelligent life?" And Professor Clarke answered, "I'm very optimistic. Someday I hope we may even find it on Earth." So the fascinating point is that for a vast period of time, from 4 billion years ago or so to about 1 million years ago, there was an enormous array of living creatures on the planet Earth which we would think were not intelligent. There were no Homo sapiens sapiens. There were first the bacteria, and then ultimately algae, the eukaryotes, and after that we had this long period of sophistication where we went to trilobites and ammonites, and then ultimately to dinosaurs and mammals. And if someone from a spaceship from another galaxy were to have landed on Earth 100 million years ago, they would have said, well, all we find there are different creatures, but we haven't been able to talk with any of them, unless they maybe knew how to talk to dinosaurs. But basically, if we had, it is only in this final thin onion skin layer of life on Earth that we have human beings capable of building spaceships and capable of sending spacecraft to other regions of the solar system, even to distant galaxies. We now have vehicles that have left our solar system and are heading out deep into space with a recording and a record showing the position of our planet with respect to the other planets of our solar system, and actually music and voices from human beings that were alive at the time this recording was made and put into the spacecraft that the Voyager spacecraft that is now heading out into the depth of space, where it may at some point in time be encountered by intelligent aliens flying in a very sophisticated kind of vehicle. But I think the best answer to your question is that life is a fundamental phenomena and may well have evolved into all manner of strange and different and beautiful and intelligent creatures throughout the entire cosmos. And perhaps at some point in time, we may get a chance to actually visit or encounter some of these creatures and learn more about life that is extraterrestrial in nature.
Thank you. And one more question, if...
okay, one other, please.
Well, I would like to know, where do you publish your papers, and other scientists who for example, we normal people can see those and trust those information.
Excellent. Well, I have, if you look on Google or Wikipedia and put my name in, you can find a listing of my papers. Many of them, many of you may be in ResearchGate. If you're in ResearchGate, I have a large number of them available for download. We published a book on an atlas of microfossils in the Orgay meteorite in 2018. I have that book on my cell phone. You can get it from ResearchGate, but anyone that would like the book or any of my papers, I have many of them here, and I can hand them to you by airdrop for free. If anyone has a cell phone and would like to get any of these, I can bring them up and turn it on to airdrop, and you can have them immediately transferred to your iPhone. It doesn't work on Androids, but AirDrop does work on iPhones. So anyone with iPhones that would like to receive those, just let me know. Is there anyone with iPhones that would be interested? In which case I can turn on my AirDrop and deliver you very quickly the atlas and any of these other works. Any other questions?
No, thank you.
Yes, right. That our origin of the life is 9.7 billion years. Yes, probably is counted there.
This paper is The Genome Increase as a Clock and the Origin of Evolution of Life. That's available on Biology Direct. I have that paper at Sharov.
And make like.
This is his calculation of bilinear regression of genetic complexity, and by taking the genetic complexity of various kinds of organisms and plotting it backward to the first life form, he is saying that somewhere in excess of 9 billion years, by this kind of algorithm, would have been the origin of the first cell. Now, that's a debatable point, but the argument is very simple. And what he is saying is that the time of origin of the planet Earth, by that point in time, there was such an incredible degree of sophistication that the actual origin of life itself should have occurred significantly earlier than that. And it doesn't really matter whether it's 6 billion years ago or 8 billion years ago. If it is longer than actually 4, then it has to have originated somewhere other than the planet Earth.
Right. And it means that our life is necessarily coming from cosmos.
What?
Our life is coming from cosmos, necessary.
It is coming from space.
From space,
yes. That would suggest that it came here from somewhere else.
No.
If this person was here, I would ask if he had one of the factorials, and the speed of the assembly of organic molecules is immense. So, so what kind of speed
well,
do you consider?
The paper is fairly complex, but I suggest that in fact, I can give you the paper by airdrop. I have it, I'm pretty sure, on my iPad, Charles' paper, and it is absolutely in keeping with the problem of the fact that the very first recognizable living organisms on Earth do not seem appreciably different than life on Earth today. Now, unfortunately, we can't do 16S gene sequencing because in the fossil record we don't have that kind of data. But we can see that clearly among the first life forms on Earth were the cyanobacteria. And they may have been delivered here from somewhere else. Again, I think the possibility that cyanobacteria developed abiotically is very, very slim. And the reason, again, is the problem of complexity of all of the genetic material, all of the biological material that must go into a single cell. You've got this amazing complexity of proteins and enzymes and DNA and RNA that is just astonishing. And at the time this theory of the origin of life on Earth was advanced in the early 1950s, one thought that basically a cell was some kind of an envelope, and on the inside you had some kind of material, some kind of genes. Genes were known, but nobody knew what a gene really was. Nobody knew what a DNA molecule really was. And nobody knew the incredible complexity of luciferase and all of these other enzymes and proteins. And it turns out that the function of a cell is so phenomenally complex, all being driven by orders being given to the cell to manufacture these products, to take these kinds of gases in and then pump those kinds of gases out, to take in the materials you want, to break them apart, put them into the various cellular components, and throw away the part you do not need. So that is the fundamental problem. We know too much, but we don't know anything. That's the problem. We know that life is astonishingly complex, and I can tell you I am absolutely certain about that. How, when, and where life originated, I have absolutely no idea. And the problem is, I do not think a human being on the planet Earth really and truly has a good idea of how, where, and when the origin of life occurred. But what I am saying is the presence of these fossils in the meteorites, as well as the presence of incredible biomolecules in various other bodies, such as the asteroids. We've now found that the Ryugu samples return does contain protein amino acids with an L excess. So that is an indication of biology on Ryugu at one point in time. And hopefully at some point we may get enough of an asteroid or enough of ices of the moon to make it possible to find extraterrestrial life. One of the most important things that the astronauts could do when they go to the South Pole of the moon is immediately examine the ice probably delivered there by comets, because if it does contain biology, that biology may still be alive, and that biology would be a chance of investigating extraterrestrial life forms in living state. And that could give rise to some of the most exciting science that has ever been obtained on the planet Earth.
Okay, I'm also astrobiologist. First of all, it is not very scientific, but I'm absolutely sure there's life beyond the Earth, and it's widespread, and it could take shapes we can't imagine. Well, that's one thing. More practical question, though, here. You spoke about the red structures on the Pluto, on the moons of the Jupiter, Enceladus, and and also on Europa. I have a question. Well, we do have organic material called toluenes on the Saturn's moon Titan. Do you think the simple spectroscopy is enough to distinguish between the toluenes on these planetary bodies and algae, which could be a complex organic organisms, not molecules, unlike toluenes? Or do we need something else, some more complex analyzing system to distinguish between them? So,
wonderful
that's
question.
Biology.
I didn't get a chance to talk about kerogen and toluenes. There are toluenes. Toluenes can be formed on ices, and there are 2 points. First, when the red colors were discovered on Europa, it was sort of hidden. Nobody really wanted to talk about these red colors. And by that I mean scientists within NASA. It was a shock. And for a long time they published them, well, they're sort of brown, but in fact they're not. They're really red. In a paper that I pointed out that I did with Sir Fred Hoyle, my late wife Miriam and I puzzled over this problem of the red colors because those discoveries had just been made, and we included a section in that paper on that phenomena. I spent a lot of time because one of the things that I was thinking is, well, if you look at where those red colors are, they're on the cracks. They're absolutely associated with the crevasses, which indicates they came up from the oceans through the crack and spread out a little bit around the edges of the cracks. So those red colors come from the oceans of Europa, not from the surface. Otherwise, they would be distributed all over the surface. So then I decided, okay, maybe they're minerals. And I spent a vast amount of time trying to find what were the red water-soluble minerals on the planet Earth. I went to the Keck Museum. I went to museums at Harvard and Oxford and in Las Vegas, Nevada, and in Los Angeles, and spoke with many mineralogists and many geologists. And you have minerals like jasper and agate and iron oxide hematite and cinnabar, mercury sulfide, large numbers of beautiful red colors in minerals, but they're all non-water soluble minerals. Out in California, in the San Francisco area, when you fly into San Francisco, you pass over very beautiful pink and red regions, and Owens Lake can be seen from space as having a huge region of red. The problem is that is bacterial rhodopsin pigment from the Dunaliella sole living in those hypersaline lakes. So we're back to biology of pigment again. So the pigment, the dominant red pigment, is bacterial rhodopsin. But again, it's made by biology. It's not made by abiotic chemistry. It's not made by mineralogy. I am convinced that the red colorations of Europa are formed by biological materials, and one of the things that I'm hoping will be possible to do is to get high-resolution infrared spectroscopy of Europa with the Webb telescope and perhaps be able to distinguish whether one is seeing bacterial rhodopsin or some other red pigments in those cracks associated with Europa. Now, if in fact it were from a dissolved substance like toluen from the oceans, that would mean that the oceans of Europa would be blood red. Otherwise, the stuff that washes up on the surface wouldn't be red. And the problem again is, you know, we have red tides and we have red seas, but again, on Earth, those are produced by biology. One of the other things, the meteorites contain kerogens. It's been known for a long time that the carbonaceous meteorite contained kerogen. Well, about 20 years ago, the scientific community stopped calling it kerogen and started calling it insoluble organic matter. And by calling it IOM, they got away from a problem, because everybody on Earth knows that kerogen is made by biology. That's how it comes from. We don't have any abiotically formed coal. We don't have any abiotically formed kerogen. We don't have any abiotically formed oil on the planet Earth. It is all made by biology. So, then the chemical composition of the kerogens of these meteorites is almost identical to the chemical composition of terrestrial kerogen. In addition to that, there was a wonderful paper last week that I attended at the World Conference on Basic Sciences and Sustainability in Belgrade on the bio production of terpenes. Well, one of the early studies of Oro and Nooner was they detected pristane and phytane in the Orgay Ci1 carbonaceous meteorite. Pristane and phytane are long-chain biomolecules that are formed by the diagenetic breakdown product of the chlorophyll molecule. Again, no one has ever found a way to produce chlorophyll by non-biological processes. So I think the toluen argument is an argument by astrophysicists and meteoriticists and planetary scientists who wish to skip across the possibility of extraterrestrial biology. So by simply saying, oh, well, it's just tolons, and yet, if you really look at the experiments that have been done, no one has ever showed us red tolons. Dark brown tolons, black tolons, yes, lots of them. Light brown tolons even. But I've never seen anyone produce red tolons or purple tolons. And if they can do it, then they should do so. And then obtain a spectra of those so that we would have a good chemical match to try to link the spectrum of the data from Europa. Again, I think Europa's material is biological.
Thank you. First off, great time. This is more of a general question I have. Let's say I'm a biology student, first year. What should you suggest? What can I do from here, Georgia, to be able to work with people like you in the future, to be able to work in NASA? If you have some suggestions for me, I'd be very pleased. And thank you.
We're getting ready to go back to the moon, and we're getting ready to go to Mars. So the first launch was supposed to have taken place yesterday, but it was delayed by the hurricane, the first return to the moon. The Space Launch System hopefully will be launched within the next couple of weeks. There will be need within NASA and ESA and the various space agencies of the planet for scientists and engineers who know all manner of fields, engineering, mechanical engineering, thermal design, rocket propellants, astrobiologists who are knowledgeable of biology, of microorganisms, of bacteria, of all sorts of different life forms, as well as astronomers and astrophysicists who will be studying the various kinds of things that we will bring back from the moon and from missions to asteroids and planets. As some of you may know, yesterday, well, I think it was actually in Georgian time last night, the DART mission impacted onto a tiny asteroid with the intention of sending a spacecraft to crash into an asteroid to see if we could actually deflect it from its path in case some of these small asteroids might be headed toward the planet Earth. Could we deflect it and get it to miss the Earth by putting in a shock wave into the asteroid? So that basically means that mathematics is profoundly important, because when you think about it, sending a spacecraft to a tiny asteroid and impacting on this little bitty body requires an incredible degree of precision in terms of communication guidance and trajectory calculations to enable you to strike an asteroid with a spacecraft, or to rendezvous with a comet, as was done with or rendezvous with asteroids like Bennu and Ryugu, or rendezvous with a comet like 67P Gerasimenko. So basically, the answer to your question is sciences, engineering, and mathematics of your choosing, and you should choose that which you will love and enjoy. I was employed at NASA from 1966 until I retired in 2012, and I don't really say that I worked at NASA, because actually I came in, I had fun, and they gave me money. So that is what you should seek in your career, something that you love doing, and you will find that you will continue to do the things that your boss will consider work, even though you might be sitting at home reading a paper or writing a paper at midnight on a Saturday night. You are still doing the kinds of things that you love and enjoy, and that's the best answer. And I think we need scientists and engineers from all over the world, and mathematicians. And I can see from the beauty and and magnificence of the architecture that I've seen here in Tbilisi, you have some really, really incredible architects and people who are doing amazing work. So that's my position on that. Thank you.
Thank you very much.
So I have the question about your research back in 2011 about the alien-like species. I would like to have more information about it and what information are in currently nowadays in 2022.
The work actually started in 1997, and it was done in a very simple kind of way. I knew in 1996 David McKay had announced the discovery of possible nanofossils in the Mars meteorite ALH 84001. He did that by electron microscopy study, and the critics came out and argued, well, what he found was not really biological, for one point, and another point was these things that he was seeing might be coating artifacts, might be the results of the gold palladium coating that he put on the sample in order to look at it with the electron microscope. Well, we had at the NASA Marshall Space Flight Center an environmental scanning electron microscope, which was different than the other scanning electron microscopes in that you could put in the vacuum chamber, instead of having it a high vacuum, you could put in a water molecules, low pressure of water molecules, and when you pumped the chamber down and you started doing the imaging, as the electrons hit the sample, water molecules would touch the sample and carry away the charge so that it would not charge up even if it was not coated and not conductive. Since they had criticized coating artifacts, I said, well, if we don't put on any coating artifacts, we can't be criticized for looking at coating artifacts. So we worked entirely with uncoated, no gold, nothing. We just broke the meteorite. We're careful to look as when it broke and put it on the scanning electron microscope stub with the freshly broken surface up, so that we would be looking at freshly fractured interior surfaces. And then simply searched for evidence of biology. And that is the procedure that I have adhered to for a long time, making sure I look at freshly fractured interior surfaces. But after the discovery in 2006 of the importance of nitrogen, I really can tell if there is something that is a modern contaminant on my sample by seeing the nitrogen content. So I really am not worried like I was when I first began this work.
We know there are prokaryotes and eukaryotes, and there are also organisms called viruses. So we know that they probably arrived on Earth 3.5 billion years ago, and there are neither prokaryotes nor eukaryotes. So, like this?
That's better, yeah.
So there are organisms called prokaryotes and eukaryotes, and there are also viruses. So they are neither prokaryotes nor eukaryotes. They're really weird organisms. So do you think they came on Earth from space? Is it possible?
I think absolutely. One of the intriguing things, you know, a lot of people like to say, well, viruses aren't really alive, but one of the problems is a lot of vaccines are made with freshly killed virus. Well, my contention is he was alive before you killed him. After you killed him, he wasn't alive anymore. So the point is, viruses, even though they are considerably simpler in those kinds of terms than bacterial cells, they still have the wonderful genetic information of RNA, or RNA and DNA, and so that this kind of genetic complexity exists within the viral world as well. And basically, they typically hijack a cell and take over the instruction set and order the cell to manufacture a lot more copies of the same virus. But we now know that there are all sorts of organisms that are viruses that attack bacteria, called bacteriophages. And so I think it would be astonishing to hypothesize that viruses as well do not exist on the parent bodies of these meteorites and did not exist within the prokaryotic and eukaryotic organisms that were found there. So in fact, there have been some papers written in which a hypothesis has been advanced that COVID-19 actually came from space and some of the other diseases. One of the works of Professor Wickramasinghe is based on the fact that back in the 1800s, several diseases occurred almost simultaneously in distantly related portions of the planet Earth. And that does provide some evidence because in the 1800s we didn't have air travel. Now with airplanes going everywhere, someone gets infected with a virus in the United States, they can be in Georgia the next day. So, but I do think that viruses could well live on the parent bodies of the carbonaceous meteorites on the comets and on water-bearing asteroids, and could have been brought to Earth at various times and triggered diverse types of plagues when a new virus arrives and humans don't have resistance to it. It could trigger a worldwide pandemic.
Thank you.
Well, no more questions. I thank you very much for your attention, and I'm delighted to have been here and been able to talk with you. Thank you.